Thank you everyone for joining this symposium. I'm Fairuz Shadmani Shishir, graduate research assistant in the department of ISRS, the University of Kansas. I'm working on AstaLab, supervised by Dr. Kamil Hawk and Dr. Sumaya Shawani. So today I am presenting my ongoing research work with MetaLLMs, residue-wise MetaLLM prediction using deep transformer model. So, what is protein? So, protein are large molecules which interact with our body and do function various stuff. So, the main function of the proteins, including growth and maintenance of our body, the causes they help us to prevent biochemical reaction for causing any harmful disease and bolster our immune health. And also, it transport and store nutrients in our body. So in the picture, you can see that we have DNA in our body, which is a blueprint of our life, and it transcribes into RNA, then it translates into protein. So protein mainly serves many purposes in our body, and they are the main machine to do tasks for various purposes, like what I mentioned earlier. So what is metal binding site? So metal binding site means a metal binding site is a single amino acid where a protein binds to a metal. So in the picture, you can see that here is a protein sequence and it's a metal like zinc. So a metal will be bind in such a position that it will be replaced by any drug or any substance. So before binding, this is the protein sequence and this will be the position. And after binding, the metal which is zinc will be bind in the position number 3. So that's the protein, that's the work of protein metal binding sites. So why this is important for the research of our ongoing AI community. So if we successfully detect the protein binding sites, the positions and the binding metals, then it will be easy for us to develop new drug or to analyze the protein better for developing our to prevent our disease or to tackle the upcoming disease with it might be numerical disease or something like that. So here is a motivational video I want to show you here. I think I cannot share it here. So what are the background of this research? So existing methods, the protein binding site methods, have low accuracy, absence of positional information, which is very important for this kind of research. And often the researchers includes a hand engineered feature, which is not good for the ultimate binding site prediction, and also predetermined set of residues and metal ions. So in this research, we want to highlight more metal ions prediction for the metal binding sites. So considering all the issue, we have proposed a end-to-end deep learning based architecture to predict metal binding sites and also metal ion. So the input protein sequence will be fed into proteins transformer model, which is a deep neural network. And after feeding the proteins model, we will have the embedding of the sequence, which will be 1,024 embeddings. And also we will feed the positional information. Positional information means where the metal will be bind in a protein sequence, the number of sequence, the length of the sequence. So then we concatenate the sequence and the protein embedding, and then fed into a feed-forward neural network to predict the metal ions. So ultimately, this will be end-to-end deep learning architecture, and we traditionally it was predicted by single metal ions, and in this work, we were able to do it multi-level metal binding ion. So like we sequentially predict calcium, copper, zinc, and many more metals in a single sequence. So here is the hyperparameter of our model, and the experiment was done on NVIDIA RTX 3090 GPU, and the training phase took 45 minutes to complete the 200 epochs, and during the test phase, it took 5 seconds to make the prediction for one batch of protein sequence. So for this experiment, we considered Uniprot database. So here there were 18,531 samples for this sequence, and the length of the sequence were between 50 to 1,000. And for the validation, we used default straightforward validation function for the 200 epoch, and the ultimate accuracy we got of this model was 90%, which is the state of art. So here are the results. I'm showing that the recall values, the F1 scores. So we did default cross validation to check where the accuracy got high. So for the iron and magnesium zinc, it got high accuracy and above 90%. And the recall value of the copper is slightly lower than the other from the other metals. And it was same for the F1 score. And it's very interesting in our experiment that the precision value to predict all the metals is very high for most of the metals. So that was a very challenging task for us to prevent to improve the accuracy, especially the precision score. So and there is a model accuracy curve. Here we got the training and validation accuracy. So we are thinking to extend our work for to tackle the future goals. So the future goals of our work will be extend our work on a large data set with a higher accuracy, and also we will develop a deep model architecture which will be considered for faster inference, because day by day the number of protein sequences increasing in the databases. So to training for large database is very challenging. We need distributed computing or lots more stuff. So it will be for our convenient way to tackle this issue and include distributed computing for developing the architecture and also faster inference. That's all from me. Any questions?
So in your proposed solution, you have mentioned the transformer networks.
Yeah.
So what it is doing and why did you took transformer? Why not any other machine?
Yeah. So especially the transformer model is a deep neural network architecture which takes the input like text or any protein sequence and give you a embedded sequence or embedded text. So for the purpose of our protein sequence, the ProTrans model is like a deep learning architecture which trained on the proteins, billions of protein sequences. That's why we chose ProTrans model to give us the embedded protein sequence.
Have you tried any other embedding methods?
Like we consider encoding and lots of stuff, but ProTrans give us the better results and better embedding.
And you said that this model is end-to-end, so you simultaneously optimize the DNN as well as the transformer network as well.
Yeah. So simultaneously we train ProTrans and the proposed architecture.
Okay. Nice one.
Any other questions?
Okay, so I have a question. As you showed us that the accuracy you were getting is about 90%. How you were comparing this? Like how do you know that 90% is a good accuracy number?
Yeah. For this specific task, we didn't compare our work with state-of-the-art models. So we will extend our works with the view to compare our work with the state-of-the-art model.
Could I ask a related question? What does accuracy mean in your model? Is it closeness to the expected protein sequence, or how do you kind of measure accuracy exactly?
So the accuracy means the predicted metals positions with the actual metal and positions.
So is it an exact comparison between the position, or is it like?
Yeah, it's exact. It was an exact comparison.
Okay.
Because if you don't know the exact position where the metal will be bind, so if the metal will bind in a different position, so the ultimate purpose or ultimate function of the protein can be changed.
I see. So it's a discrete position.
A discrete position.
Okay, thank you.
Like the numerical position of the sequence.
I see.
So any other questions? Yeah, go ahead.
No.
Okay, any other questions?
No.
Okay, yeah. Thank you, Fairuz. And just fill out the grading rubric, then after a couple of minutes, we can start the second presentation. Thank you.